are an iReach organization and the RL team meeting today. A few reminders. Number one, CME credits can be obtained if you've not registered. You can contact Deborah Reese by email and she'll facilitate that. Number two, we have a tech talk on November 7, I believe. Jessica Webster from the California Department of Public Health will be talking about C. difficile disease, a disease I know and love well, oddly enough. And we'll be discussing patient risk factors and healthcare facility issues. And also for your calendar, December 17th, we have another innovative discovery series with Julianne Trabolsi. She'll be discussing infant dietary needs and outcomes. Today's presentation is going to be excellent. Dr. Sims-Mourtada presents today. She currently functions as the Associate Director of the Cawley Center for Translational Research. And under that umbrella, she is the Director of the Breast Cancer Translational Research Unit. She has come to us from UT, where she did her undergraduate, her PhD, and her postdoctoral training. And we're fortunate that Dr. Petrilli was able to recruit her here in 2011, where she's now a clinical scientist as well. She has significant biomedical skills, and you'll learn that today, I'm sure. And she has funding from Department of Defense, from NIH, as well as a mostly funding, extensive publications, reviews for multiple publications as well, and continues to train postdocs and surgical fellows. So we're really fortunate to have her here in this organization. Her topic today is triple negative breast cancer, a disease which is quite aggressive, and I didn't know that Delaware had a very, very high incidence of this. Aggressive disease, widely often metastatic, tends to affect young women and women who are Black as well, more in that direction. She's going to talk today about disparities in this disease state, social parameters, as well as biomedical markers and issues. And looking forward to this presentation. Dr. Sims-Mourtada.
Thank you very much, and thank you for having me here today to share some of the work that we're doing in the CTCR. Just share my presentation. So can everyone see my slides?
Yes, it's fine.
So just a little bit outline of my talk. I'll give an introduction to the Cawley Center for Translational Cancer Research, in case you're not aware of it. We'll talk a little bit about breast cancer and particularly breast cancer disparities in Delaware. And then I'll talk about some of the research that's going on within our breast cancer research program to address some of these disparities. So the Cawley Center is a 7,000 square foot lab space. We're located on the 4th floor of the Helen F. Graham Cancer Center. We are one of 5 wet labs that exist within the country, within a community cancer center. So that makes us pretty unique, and we are immersed where the majority of cancer patients get their care at a community cancer center. So that provides a lot of advantages for our research. The Cawley Center has research in colon, breast, and ovarian cancer right now. We have several core facilities, including flow cytometry, tissue microarrays, tissue procurement, and I'll talk a little bit about this later. We've just started an organoid core. And as Dr. Bacon said, we support training for undergraduate and graduate programs for the University of Delaware. We provide the majority of training in cancer research for the university. And our mission is really to establish a translational research center of excellence that uses multidisciplinary backgrounds to discover new cancer treatments, prevention strategies, and improve the lives of cancer patients, and of course, reduce cancer disparities in underserved communities, which is a mission of ChristianaCare as well. We have basically 4 main goals for the Cawley Center. The first one is really fostering this collaboration between clinicians and scientists for true bench-to-bedside research. We have collaborations with a lot of our breast surgeons in my group. We also have a research consortium with the Wistar Institute which brings scientists from a basic science institution to collaborate with our clinicians at the Cancer Center. Another goal of ours is addressing health disparities, which you'll hear a lot about in my talk today about some of the ways we're going about that. Maintaining state-of-the-art research infrastructure. We do that here at the Graham Center by purchasing new equipment and also through our partnership with the Wistar, which allows us to access core facilities with the latest technology that's available for research. And a major goal of ours is really training the next generation of scientists, undergraduates, graduates, and we also have postdoctoral fellowships. And just kind of a shameless advertisement right now, we're actually recruiting for one of our fellowships, the Lisa Dean Mosley Fellowship in Cancer Stem Cell Research. This is available to PhDs 1 to 3 years out of their graduate degree, or for residents within 1 to 3 years of completion of their medical degree. So if you know anyone who may be interested in applying for this, the job is actually posted right now under Research Resident, or they can send an email to ctcr@ChristianaCare.org. So the Translational Breast Cancer Research Program was started in about 2014, I believe, as a collaboration between myself and members at ChristianaCare, University of Delaware, and Wistar. We have quite a range of expertise, from clinical expertise. We work a lot with chemical engineering department, biomedical engineering department at the University of Delaware, and then a lot of scientists from Wistar. One of the unique things that we have is a community research advisory board that supports our research. This includes breast cancer survivors, primary care physicians, because they are really the ones who first see these women when they come in with breast problems, and also local community leaders that we look to help us solve some of the disparities that we see going on within the community. Our areas of research focus are really on improving breast cancer prevention and diagnostics, developing new treatments for metastatic and triple-negative breast cancer, and of course, decreasing breast cancer health disparities. To talk a little bit about breast cancer in Delaware, we're ranked 12th in breast cancer incidence, and our breast cancer incidence is growing in Delaware and is significantly higher than the US rate. We're ranked 15th for breast cancer specific mortality. And breast cancer is the second most common cause of mortality among all females in the US and in Delaware. And it is the #1 cause of mortality among Black women with cancer mortality. And so to kind of go deeper into disparities in Delaware, non-Hispanic Black females have a higher incidence rate compared to non-Hispanic white females. They're also more likely to die from cancer. Across the US, about 40% more likely to have breast cancer specific mortality. And that is seen in Delaware as well, as they have a higher mortality rate compared to non-Hispanic whites. This is particularly evident in younger non-Hispanic Blacks, which have a higher age-adjusted mortality rate than younger non-Hispanic whites. And they, along with Hispanic Black females, along with Hispanic females, are more likely to be diagnosed at regional stage or later compared to white females. However, when we look at our mammographic screening rates for Blacks and whites, they're really not statistically different. And this is across the board, looking at total populations of Blacks and whites. I know some of our population health team is going a little bit deeper in this to see if there is a disparity among particular groups that are not getting screened. But it does seem to be something more than just finding these cancers early. We think that there is a biological component that is leading to these disparities between white and Black women. And part of that is due to triple-negative breast cancer. As Dr. Bacon said, this is an aggressive form of breast cancer. It more often affects younger women and Black women. It's a rapidly growing cancer. It passes very quickly through the in situ stage, and patients are more likely to have early metastasis within 5 years of diagnosis. And unlike the other types of breast cancer, we currently don't have a good targeted therapy for triple-negative breast cancer. Immunotherapy works in some cases, but it's really only around 20%. And as Dr. Bacon said, the state of Delaware is ranked among the states with the highest incidence of triple-negative breast cancer. This is some work that we did based on our Delaware State Cancer Registry, looking at incidence rates, which are pretty stable and higher than the national average of about 22.7 per 100,000 women, based on the national average, which is around 15. So definitely higher incidence of triple negative breast cancer. And a report that just came out last year actually shows that we're not only the highest in incidence of triple negative breast cancer, but we have the highest disparity in triple negative breast cancer. So when broken down by race, we actually rank around 14th in white women, which is about the same as our incidence of breast cancer overall. But we rank #1 in incidence of triple negative breast cancer in Black women. And as you can see here, this is incidence rate per 100,000 for non-Hispanic white women versus non-Hispanic Black women, and Delaware is up at the top. So we do have a major issue in triple negative breast cancer and the largest disparity in the US. So why are Black women more likely to get triple negative breast cancer? We believe it's a combination of factors. It's been linked to socioeconomic factors such as low income, poor diet, environmental factors such as pollution. There are genetic factors, increase in oncogenes, BRCA1 mutations, the Duffy null gene, which is a chemokine receptor that's located on red blood cells. It's believed to have a selection effect for this gene because it helps to reduce inflammation during malaria, which is very beneficial. But for breast cancer, it's not necessarily actually drives chronic inflammation. There are also other breast cancer risk factors that we know occur more often in Blacks, such as metabolic disorder, parity without breastfeeding, and alcohol use, which we'll talk about later on during the talk. But one thing that all of these factors have in common is chronic inflammation. And so we believe that having this chronic inflammatory state at the time that you would get a mutation can actually promote triple negative breast cancer. And we know that chronic inflammation increases proliferation, angiogenesis, and immunosuppression, but it can also induce mesenchymal phenotype, which is a feature of triple negative breast cancer, and increase stemness within the tumors, which is another feature of triple negative breast cancer. So we're currently trying to understand how chronic inflammation, both systemically and at the tumor microenvironment, might influence growth of triple negative breast cancer. So my group really studies the tumor microenvironment in triple negative breast cancer. As we know, breast cancer doesn't exist just tumor cells by themselves, but it's surrounded by a lot of other cells, immune cells, stromal cells, endothelial cells. And so these all interact with each other, and there can be certain growth advantages or disadvantages depending on these interactions. So triple negative breast cancer is really a heterogeneous disease, and the other cells that are in their microenvironment vary from patient to patient, but they can also vary within the same tumor. So this is a tumor section that we are currently doing spatial transcriptomics on, so we can actually sequence cells from areas, different parts of the tumor that have different types of cells there. So we see the tumor cells stained in green, stromal cells stained in yellow, and the immune cells stained in this pinkish color. And you can see that even within this one tumor, you have areas where there are high levels of infiltrating lymphocytes versus areas where there are lower levels of infiltrating lymphocytes. And so we're trying to understand what the differences in gene expression and treatment resistance we might see in these different areas. One thing that we know, thinking about racial differences in the tumor microenvironment and how that might be linked to disparities, is that Black women with triple negative breast cancer have tumor microenvironments that appear to be more inflamed, having more signs of a chronic inflammatory response. So both white and Black women have immune cell infiltration in their tumors. And for white women, this is prognostic of good outcomes. But for Black women, it is really not prognostic and can actually be, in some cases, prognostic of poor outcomes. So we're trying to understand the differences in this, and this is most likely linked to differences in the type of immune cells that are entering the tumor. We know that Black women, for their T cells, intratumoral T cells, they have high expression of immune checkpoints. And they also have some different types of cells there. They have lots of M2 macrophages, which actually secrete growth factors that promote tumors. They have immunosuppressive myeloid cells, and T cells. They have an increased microvasculature, which is probably due to factors released by these chronic inflammatory cells that promote vasculature to help the tumor survive. And they have a cytokine profile that resembles chronic inflammation, particularly a Th2 profile. So there is some evidence kind of linking these risk factors that are known to induce chronic inflammation and the chronic inflammation that we see in the tumors of Black women. This is a paper that my group published last year, looking at interactions between tumor cells and B cells. And what we found is that the tumor cells can actually direct the B cells on how they're going to respond in their immune response, causing them to make an immunosuppressive antibody that's not effective for stimulation of immune response against the tumor. And this is driven by a Th2 cytokine called IL-10. And we weren't expecting this, but we found in this study that Black women actually had 2.5 times higher amount of IL-10 within their tumors. And high tumor expression of IL-10 was associated to poor outcomes and this IgG4, which is an immunosuppressive antibody response. We're still trying to understand what's driving IL-10 expression in these tumors in hopes that we can actually find a target for treatment for women who have these IgG4 cells in their tumors. Another risk factor that we're looking at that's linked to inflammation is alcohol use. A lot of people are aware that alcohol is a risk factor for GI cancers, but it's not well appreciated that it's also a risk factor for breast cancer. And light to moderate alcohol consumption can actually increase breast cancer risk for about 12 to 30%. And so that's a lot of women when you think about, greater than one drink a day. It was originally appreciated that alcohol is a risk factor for cancer because its metabolite, acetaldehyde, is a mutagen and can introduce mutations into the different tissues. But we've recently discovered, particularly for breast cancer, that there are other mechanisms of alcohol that also lead to cancer risk, such as altered estrogen levels and an increase in blood acetate, which not only promotes tumor growth, but also can provide immunosuppression so that the immune system is not able to respond to the mutated cells. And one factor that we're really looking at in racial disparities is alcohol-associated inflammation. So another thing that we are number one at is alcohol-induced breast cancer. So this in Delaware. This was a paper that was published in Cancer Epidemiology in 2021, where they looked at alcohol-attributable breast cancer cases. Delaware had the highest incidence and the highest amount of cancer deaths attributed to alcohol. And it was estimated that around 20% of breast cancer cases were attributable to alcohol in Delaware. So this is something that our community board is really trying to disseminate the information that alcohol use can promote breast cancer. When we look at alcohol and breast cancer disparities, we see that traditionally Black women had alcohol use, used alcohol at a much lower rate than white women. But the alcohol use in Black women is increasing now at a faster rate than white women. And low-income Black women have higher rates of binge drinking compared to their white peers. There was a large study called the Carolina Breast Cancer Study that looked at alcohol use in thousands of women that showed that alcohol use increases the risk of triple-negative breast cancer only for Black women, not for white women. And we think that this is due to the inflammation that's induced by alcohol. There was another study that came out that showed that there are racial differences in inflammation in Black and white women, that Black women have more immunosuppressive Th2 type cytokines than white women after alcohol use. So the major metabolizer of alcohol is this enzyme called aldehyde dehydrogenase. Aldehyde dehydrogenase, there is a family of enzymes. A lot of them are involved in alcohol metabolism, but they are also involved in metabolism of intracellular and extracellular aldehydes. And my group has been studying these enzymes for years in the context of cancer stem cells. So Aldehyde dehydrogenase is a marker for cancer stem cells, which are resistant cells in cancer. And we've known that it's important for cellular differentiation of cancer cells, but also for immune responses. And it's recently been shown by my group and others that expression of one of these isoforms, ALDH1A1, is induced by chronic inflammation, and it's also induced by alcohol metabolites. And so we think that this chronic inflammation is upregulating this enzyme, not just in the liver to improve alcohol metabolism, but it can be upregulated in the breast and other tissues as well. And it's been shown that this enzyme also regulates visceral fat and glucose tolerance, so obesity and diabetes, which are also risk factors for breast cancer. And so we think that this might be a link between chronic inflammation that may be induced by alcohol, but also the link between chronic inflammation, diabetes, and breast cancer and obesity. So it's become a very important target of interest for us to see if we can actually target this enzyme to improve outcomes. My group published a study last year looking at expression of ALDH1 in triple negative breast tumors. We looked at, I believe, almost 200 cases, and we saw that expression or high expression of these women is found more often in Black women, women who are obese, or women that have prediabetes or diabetes. So supporting the hypothesis that ALDH1 may be the linker between these different risk factors. And when we looked at outcomes, we see that women that have strong expression of ALDH1A1 in their tumors have lower recurrence-free survival and also lower overall survival. But when we broke it down by race, we saw that Black women who have high expression have worse outcomes than white women that have high expression of this enzyme. Where if we looked at women, Black and white women who have moderate or low expression of this enzyme, there was no difference in outcome. We're still trying to understand this and to understand if ALDH plays a different role in Black women versus white women. We're currently looking to see the role of ALDH1A1 in the immune microenvironment of breast cancer. A previous study had shown that when you have higher levels of ALDH1A1, you also have higher levels of the NF-kappa beta pathway, which is a pathway that activates gene expression, driving chronic inflammation. And so my PhD graduate student, Grace Ajay, has generated CRISPR-Cas9 knockout clones of ALDH1A1. And we see that within the two different tumor cell lines, there are different functions in several different areas, but this is a Western blot showing phospho P65, meaning activation of the NF-kappa beta pathway. And so you can see in a couple of these clones, when you lose ALDH1A1 expression, you have less activation of NF-kappa beta signaling, indicating that ALDH1A1 is driving some of this chronic inflammatory response by the tumor. My postdoc, Mosley postdoc, Iwi Ma, is currently looking to see what types of inflammatory cells are surrounding these ALDH1A1 high tumors and how this might relate to tumor immunity. So she's just started this study, and we haven't really had any real analysis, but preliminary studies suggest that ALDH1A1 high tumors might have a lot of these infiltrating lymphocytes, which we need to further characterize to see if they're immune activating or immunosuppressive. One of the issues that we have with studying some of these markers, such as ALDH1A1, in our models, is that we really do not have good models for racial disparity. And this is a paper that came out in 2022 that reported that the majority, almost 80 to 90% of our models, come from women of Western European ancestry. And so it's become one of our goals to develop models to study some of these markers or genes that we believe are involved in racial disparities, that we have appropriate models to study them in. So one of our goals is really to create these diverse models. There are two approaches. One is organoid cultures, which we are starting to collect and form an organoid core. The other way is by humanized PDX models, which are being done by our colleagues at the Wistar Institute. So having primary patient samples instead of cell lines to study different drug response or roles of different genes is very important for us, because not only can we generate the models to do drug screenings, but we can also get the data from the patient so we understand the factors surrounding this patient, such as comorbidity, environmental risk factors that they might be exposed to, and we can also collect data on ancestry. So as I said, we started a core for this at the Cawley Center. Organoids really have a lot of advantages as they come from primary tumors. So this is how we grow them up. So you can see these little clusters of tumor cells that start to form like the tumor organ in the body. And we can also introduce other types of cells into the tumor to make up the tumor microenvironment. So this here is depicting lymphocytes going into the tumors. This is really important if we want to study interactions between tumor cells and a specific type of cell. That this really can't be done very well in animal models. So we look to our organoids to understand the interactions between all of these different cells within the tumor microenvironment. This is just a schematic about how organoids are prepared. So we take patient tissue, we dissociate it into single cells, filter out all of the debris. These cells are then put into this dome, which contains something called matrigel, which is made up of extracellular matrix proteins. And so this provides an area for the cell to attach and to be more like it is in the human body versus when it's attached to plastic, which is what we've been studying for many years. So we believe that this is a much better way to get real data that more closely resembles what's going on in the body. These organoids are then grown, and then they can be dissociated and replated for another study, or we can bank them and they can be used by many different investigators. And so one of the advantages to this is that we can take a tumor from one patient and we can actually split it among a lot of different studies. So this is really advantageous for breast cancer, particularly as cancers get smaller or patients go for neoadjuvant chemotherapy. We don't have a lot of breast tissue to access for our studies. This is just some images of our organoids. So we can do this in normal breast tissue. We see what resembles ductal formation within the normal breast. Excuse me. And this is a triple negative breast cancer organoid. These little dots here are actually lymphocytes that we introduced into the organoid. And you can see they kind of form these little tunnels where they migrate towards the organoid and begin to proliferate at this site. And this is a little bit more detailed study. So we're interested in, why some lymphocytes remain outside the tumor cells and kind of cluster there versus in some tumor sections, we see that these cells actually go into the tumor. And so we can take these lymphocytes, in this case, it was B cells, put them into the organoid and trace which ones actually go in. As you can see here in red, these are the B cells staying with CD20. They're actually not just staying outside the organoid, but going inside. And we're interested in looking at interactions between these cells and the tumor cells. We can then section these organoids and look for different markers in response to treatments or to benchmark against an H&E slide from the original patient to see if we're seeing the same phenomenon occur in our organoids compared to what was actually taken out of the patient, kind of as a QC procedure. One of the things that we're interested in is doing these drug screenings. So these are diverse organoids that might respond differently to different drugs. And so we really want to introduce this into preclinical research for our own research, as well as other research that people are doing to develop targeted therapies for triple-negative breast cancer. So we've developed a couple of assays to be able to trace response. One is an Alamar Blue readout, which is based on cellular metabolism. So this chemical undergoes color change if the cells are viable or not. And we're able to trace this color change to measure cell viability. We can also do organoid forming assays where cells are treated, and we can count how many organoids grow up after treatment with different drug concentrations. And we can also look at live dead cells with staining of different biomarkers. A collaboration that we have with the University of Delaware, Dr. April Clarkson, is really trying to more authentically model inflamed breast tissue that we see, particularly in tumors from Black women. And so April has developed these extracellular matrix proteins with modifiable linkers, so we can put in different peptides into the matrix, and we can use this to put in different cytokines or growth factors that we see in inflamed microenvironments. And we're also working with Inventia, and April's group has a strong collaboration with them to do 3D bioprinting of these organoids. And this is really good because we get reproducible organoid formation, and we can also print different types of cells next to each other. So remember in the triple negative tissue, I showed you where we had a region that had lower lymphocyte infiltration versus higher lymphocyte infiltration. We can mimic that in the organoid depending on how we print these cells. So this is an ongoing collaboration. We just sent an R01 to be able to grow these tumor cells from Black and white women in these different inflamed microenvironments and do drug screening based on if the microenvironment was inflamed or not, to see if we can more authentically replicate what might be happening in the patients. The next project I want to finish up with is called the Disparate Project. This is a collaboration with Dr. Scott Siegel, who is Director of Cancer Control and Population Sciences at the Graham Center. It's called Disparate because of the name, detecting epigenetic signatures to prevent avoidable racial inequities in triple-negative breast cancer. And this was funded by an Embry Women's Health Supplement Award. So Scott has been working for several years looking at incidence of breast cancer and breast cancer subtypes in our catchment area, which for the Helen F. Graham Cancer Center is mainly Newcastle County. And what Scott has showed is that there are a couple of areas we call hotspots, notated here by the red lines, that have a higher incidence of triple-negative breast cancer versus other subtypes of breast cancer. So not incidence of breast cancer in general, but higher incidence specifically of triple-negative breast cancer. And he's also found some areas that have lower incidence of triple-negative breast cancer. And then he went on to show that these areas of triple-negative breast cancer have high exposures to breast cancer risk factors. So he looked at metabolic disorders, environmental exposure such as pollution, parity without breastfeeding, and alcohol use, all of which are risk factors for triple-negative breast cancer. And he showed that when you have cumulative exposure, so exposure to more than one of these risk factors, the more risk factors you have exposures to, your odds of triple-negative breast cancer go up. And then he overlaid these exposure scores, and we see that the regions that have higher levels of triple-negative breast cancer have the highest cumulative exposure scores. So this kind of supports our hypothesis that these exposures are driving this high incidence of triple-negative breast cancer. And we wanted to see, can we find evidence of changes in chronic inflammation in women who live in hotspots and women who don't live in hotspots? We are not the only ones to associate inflammation to neighborhoods. This was a study that came out in 2022 that showed that women who have a higher socioeconomic status and live in areas that have more what they call greenness actually had lower systemic inflammatory biomarkers than women who lived in more city type environments with lower socioeconomic status. So we wanted to look at a particular pattern that we know is changed in immune cells by chronic inflammation, and this is an epigenetic change controlled by methylation. So methylation is a methyl group that's attached to DNA that changes the way that genes are expressed. And we know that this is modified by chronic inflammation, but not acute inflammation. So we think that this might be a good way to track exposure to chronic inflammations, and potentially could be a biomarker for triple-negative breast cancer risk. This is based on some previous study that I have done looking at epigenetic changes in breast cancer in general. And in this study, we were able to show that we can actually pick up epigenetic differences in women who have breast cancer versus women who do not have breast cancer. And we could actually use this to predict if a woman had invasive breast cancer with a 70% accuracy. So this was looking at all tumors and sites that predict women who have normal breast tissue versus invasive breast tissue. But in this study, we really wanted to focus in on triple-negative breast cancer and also what's going on in these hotspots. So this is kind of our outline for our hypothesis for the study. Linking neighborhood factors, which we call the exposome here in blue, to individual factors. And we hypothesize that this drives a change in epigenetics of the immune system, which drives chronic inflammation and also this Th2 immune type response. And this can help to promote growth of triple-negative breast cancer. So we did a pilot study, very small study, looking at 32 patient blood samples, 16 patients from triple-negative hotspots and 16 patients from non-hotspots. And these patients were matched for race and stage. We then did a targeted next generation sequencing panel that sequenced around 4 million methylation sites, and then looked at which sites were differential between the hotspots and non-hotspots. And this work was done in collaboration with the University of Delaware Bioinformatics and Computational Biology Core Facility, particularly Madeline McDonald and Sean Polson. And what we found is that the epigenetic patterns from women within these hotspots and cold spots cluster together. As you can see, the hotspot clustering here, the cold spots clustering here. And there were several thousand differentially methylated genes between these two groups. We still do see some clustering by race. That was expected, but there's a clear statistical difference between women who live in hotspots here in orange versus women who live in cold spots. And so this was women with diagnosed triple-negative breast cancer. We also see this effect in women who have diagnosed hormone-resistant positive breast cancer, although it's not, the clustering is not so good as it was in triple-negative breast cancer, but there does seem to be a neighborhood effect on epigenetic patterns in these women that is statistically significant as well. So we want to expand this study to really understand this, to understand the role of these epigenetic changes on outcomes. We are not sure about the hormone receptive positive patients because we're only looking at these immunohistochemical biomarkers, not at gene expression. So we know that a subset of the HR positive breast cancers actually on a genetic level match a basal-like phenotype that's seen in triple-negative breast cancer. So we need to expand this study and kind of dig a little bit deeper into the site changes that are going on and if they're related to immunosuppressive phenotypes in these women. But our overall goal really is to kind of rethink how we do breast cancer screening and prevention. We know that there are a lot of modifiable risk factors for breast cancer, but we don't really have a good way to follow these. We have really good tests for hereditary cancers, but not for risk for other types of breast cancer risk factors. And so our goal is to develop a blood test that would be able to predict risk for breast cancer, particularly triple-negative breast cancer, because these factors are modifiable and there are interventions that can be done. And we know that epigenetics is modifiable itself. So once the stimulation is removed, the epigenetic profile can return back to baseline. And so we think that we can develop these epigenetic biomarkers to follow some of these breast cancer risks. And you can kind of think about it of how we use cholesterol to follow risk of heart disease. So that's kind of how the approach we're taking and working toward that goal. And so with that, I will be happy to take any questions. I'd like to thank the members of my lab and the research coordinators, my funding mechanisms, as well as my collaborators, both at ChristianaCare and UD, and all of the women who have donated blood and tissue samples that make our research possible. So stop sharing.
Thank you.
And I'd be happy to take any questions.
I'm sure we'll have some, and thank you very much. For questions, please raise your hand and we'll get to you in an organized fashion. I do have two questions, if you don't mind starting off. If chronic inflammation is a driving issue here, what's your experience epidemiologically looking at other diseases that cause chronic inflammation? For example, HIV treated, untreated, number one, and number two, rheumatologic diseases with chronic inflammatory process, hepatitis C. Are there different pathways for inflammation that wouldn't be associated with those disease states?
So in some disease states, there would be a linkage, like in cardiovascular disease. It does seem to be some overlap with the inflammation that we see in breast cancer, certainly diabetes and obesity. Rheumatoid arthritis, it's a different type of inflammation. It's a Th1 response versus the Th2 response that we see. So we don't think that chronic inflammation causes the breast cancer. We think that chronic inflammation drives what type of breast cancer that you get.
Okay. Thank you. And another question, the work you and Scott did with the geography, these are young women getting this disease. And so how do you correct for the fact that if they're 30 years old, for example, they've spent 2/3 of their life in a different location, growing up with different risk factors that you can identify geographically? Are you able to correct for their locations and their risk factors from prior sites?
I believe Scott is working on that, trying to determine, the time of residency. That's a hard thing to do at a population level. So yes, that is a limitation to the study right now, is that we don't know how long these women have been here. We also know that the exposure time, usually around puberty, to your mid-20s, is when we think that a lot of these exposures occur. So it's not necessarily where you're living right now, but where you were living then. And so that's also really, really hard to trace.
Yeah, no, but that's really interesting. And that whole discussion supports the fact that Delaware is a great spot to investigate these things in terms of ability to access data like that. I think we're really very lucky that way. I'm going to stop holding people up. If there are other questions, I'll be happy to direct them to you. Rashada Dawson has a question.
Hi. Hello, everyone. Hi. It's
Hello.
So I just wanted to clarify what I saw on your first slide. Did you say that the risk factors decrease if a woman has breastfed?
Yes.
Okay.
And that's because breastfeeding is thought to decrease the inflammation that occurs. It kind of prolongs the time of the breast. Breast cells expand right during pregnancy, and they undergo death thereafter. But if you're breastfeeding, it kind of slows down that process, and so you don't get this inflammatory response that you would get with breastfeeding. And it can also alter some hormone levels as well, and that's thought to contribute to some of the protective effect of breastfeeding.
Wow, thank you. That was very informative. Thank you.
I'm sorry, I think Valerie has a question.
Hi, yes, thank you. That was a great talk, Dr. Sims-Mourtada. So as you're working on sort of fixing some of these epigenetic factors, should we on the clinical side be counseling people differently as to risks, for example, with alcohol based on demographics tomorrow?
Yeah, well, I think that's a question for Scott of where the demographics for alcohol actually are right now. But I think that's where we're going, to really take from a population health level based on the neighborhoods that you live in, your breast cancer risk might be different. And so I think in a few years, we might be taking that approach where we consider where the person lives or has lived and some of their lifestyle factors to create a better risk calculator for them.
Nice. Additional questions?
Jennifer, there's a question in the chat box from Kristen Kelly.
Oh.
I can't get it to open.
I'm trying to get, oh, here we are. I had it open. Let me just check it again. Thanks, Deborah, for that. Does the usage of GLP-1 receptor agonist to decrease inflammation possibly impact the incidence of triple-negative breast cancer?
So I'm not sure about the incidence. I don't know if that's been studied. I know it's been tried as a treatment, and I don't think it's been too successful, although I think that there might be still some ongoing trials with it with triple-negative breast cancer, but I'm not aware of people looking at it as far as the incidence of triple-negative breast cancer. That's a good question.
Other questions? I do have a question in general. In the HIV world, disparities arose in multiple different populations early on, some not driven by risk factors as much as late presentations of those risk factors, coming in later than usual or more advanced. Has that been an issue as well in this setting?
It's definitely an issue for breast cancer, and lack of screening or lack of, once you're screened, getting in for treatment is an issue. But we don't think that accounts for the entire issue. We do think that there's something biological going on.
Yes, well, I appreciate that. Thank you. Other questions? Any further comments? Because frankly, this was an excellent presentation. It's just well received and wonderful. I really enjoyed it, actually. Good. Well, thank you. With that, Deborah, there are further discussions today on the agenda?
That's it. Thank you so much, Jennifer.
Dr. Sims-Mourtada, that was wonderful. Really good. Thank you. And thank you all for attending.
Thank you. Goodbye. Have a good day.